My name is Vinit Saxena, and I'm the presenter today. I have the real distinct privilege of presenting to you our fifth webinar, and it's titled 7 High-Impact Process Choices that you can use to reduce cycle time, reduce FTEs, reduce facility size, and reduce cost. I know those are very, very important factors for all of us who are developing new processes or putting up new plants. And so in this webinar series of smart processing, we've looked at various areas of reduction of complexity, reduction of footprint of the plant, reduction of cost, of course, reduction in time. So we launched right into it. Our webinar is going to be about 45 minutes. We'll have a few minutes for Q&A. You can certainly put in some of your questions in the chat. You'll see some people on this who've joined via Zoom, but the majority, because this is being broadcast on LinkedIn, we have over 200 signers on LinkedIn where it's been broadcast. So it's not just a few of you, but know that there are several other people. The only thing is you guys have the ability to actually ask questions in the chat, whereas for the rest of them, it's just a broadcast. They can view it. But I will encourage any of you, those of you who are joining us directly on LinkedIn, to reach out should you have any questions, okay? And we'd be happy to do a one-on-one and talk further. And without further ado, then we will get started. So just a little bit for some of you who are joining us, they may not know who Sepragen is, right? So I'll just take a minute to establish that background so you understand who we are and what gives us the ability to talk about these issues. And so firstly, we are a company that has been in existence for, well over 35 years. And there are almost 40 FDA approved drugs that are made using our platforms, whether it's columns or chromatography systems, like that. And there are also 2 major COVID-19 vaccines commercially available, not something that somebody developed in a lab or wasn't approved, but these are actually vaccines that many of you got during the COVID era. And there is in excess of $20 billion worth of these drugs and vaccines that are produced using our platforms. And so what are some of our equipment? Well, you learn more about it over time. If you're interested, you can certainly contact us. We are original inventors of novel column design called radial flow chromatography, which can really be used for all your chromatography needs with the exception of size exclusion, right? So it's a very unique column that enables high throughput. We have a whole line of chromatography systems and skids. People don't know about this, but all the way from bench scale to large scales. And we also have single use, which is disposable flow path systems and skids from the smallest of scales used in the biotech industry, primarily for gene therapy, to very large scales. And then we have continuous chromatography systems. And then, of course, inline buffer dilution systems, so you can reduce the quantities of your buffers that I use, which other people also have, and also inline what some folks call conditioning, but we call stock buffer blending, where you can use individual chemicals or the constituent solutions of the buffers and make single strength buffers in line. And then very interesting, very unique, concatenated unit operations where we've actually integrated many of these unit operations into single skids, and we are providing customized solutions. So should you have, we know that everyone's plant and process is unique, and so should you have those kinds of situations where, a single box solution from another vendor may not fit your needs, give us a call. And then with that, I'm gonna now transition over to our talk for today. And this is just showing you that we have these systems and equipment for different scales. Okay. So what is the root cause of poor workflow, which means low cycle times, very high FTEs, big plant sizes, and poor plant output. So the process choice and the equipment selection all combined together really are the root cause of some of the issues that we face. And so when we just need another piece of equipment or need to develop a process, very often we say, oh, there's a platform process, let's adopt this, cut and paste and run with it. It doesn't necessarily bode well for everyone, right? You may have facility constraints. Your facility may be designed in a certain way where that same process may work well for someone else, may not work well for you, because it may not have as big a buffer room, or your purification suites might be smaller. What happens when titers go up? So there are lots of issues around that. And so, of course, the first thing is that our traditional process workflow in the bioprocess industry, even for well-known molecules, is pretty complex. It's slow, it's labor intensive, and of course, that means it's costly. So if you look at what are the cost contributors to the cost of making a biological drug, of course, amortized development costs, right? R&D costs, development costs, we got nothing to do with it. So that's not actionable. But there's amortized capital costs, which is the capital cost of all the equipment and the buildings. That's actionable. Of course, if you design your processes smarter and better, you can have power-packed, smaller facilities. Consumables, again, if your process is smartly designed, for instance, you may not have a lot of cleaning, perhaps those cleaning chemicals could be reduced. And then again, if you don't have so many unit operations, the labor costs could be reduced, right? So we have here, this is just taken from literature, and what I'm doing here is providing perspective that other people in the industry have encountered and have talked about, right? And of course, the slow traditional batch processes, whether they're batch or fed batch, have been slow, right? So the cycle times are long. Why is it? Well, you do cell culture, you do centrifugation or filtration, you capture chromatography. This is for a MAB line, right? So you're just using it as a model, but it could be different for fermentation, it could be different for gene therapy, sometimes more complex even for gene therapy, even though the processes are smaller. Centrifugation, capture, viral inactivation, flow-through chromatography, UFDF, formulation, fill, and finish. And then in between these steps, there are hold steps. And so, we are waiting to see how things went at that step, and then we move on to the next and the next and the next. And forgotten in all this are ancillary activities, right? And really, you'd call them, they're not so much process unit operations, but they're ancillary operations like buffer management, right? You've got to make buffers for all the chrome systems. If you're doing UFDF or doing a buffer exchange, again, you need to make buffers. So all that, and that has to be QC, right? So columns have to be packed. So things like that, all those kinds of essential operations are often forgotten and become an afterthought when the process train is being designed or a facility is being designed. So in this case, for instance, you have you could do cycle times of 8 to 10 weeks. You have a $200 million facility, a large facility that takes up to 2 years to equip, right? And even though we talk about basic unit operations, you look at buffer blending, right? Making buffers. Bioforum did a study and they concluded that 2,000 liters of buffer will take about 7 hours and 2 FTEs. Well, that's staggering. That's staggering considering how much buffer we actually use in a process and not only do you have to make buffers, if you're making buffers, let's say the day before, or the night before a campaign starts, those buffers have to be stored somewhere. And those totes, as many of you are aware, then line up. If you don't have a storage space, like in this particular diagram here, floor plan here, this pink or this maroon area is all dedicated to buffer storage, right? So huge amount, buffer prep, buffer storage. If you have in this case, it's a ballroom type process facility, right? And so if you can eliminate that, potentially you save 30% of operational square footage and FTEs devoted to all that, right? And of course, it adds to your cost of goods. Of course, there are QC issues that come with each of those totes that are filled and waiting to the next day or two. How do you ensure that the pH, conductivity, et cetera, of those solutions remains the same, right? So, and then as the titers go up, what happens? We are living in an age where, our upstream folks have done a superb job of increasing titers, right? 4 grams a liter is normal. So older facilities that were designed for low titers, obviously we have an issue, right? The purification suites were small because smaller columns were needed. The buffer making capacity was low because less buffers are needed. And now those are bottlenecks. So the standard challenge to these kinds of capacity increases downstream is simply, oh, let's get more columns. Okay, titer's gone up. Let's get more columns. Let's get more skids. And it's not that simple, right? Because then you're up against facility constraints, you're up against manpower or human power constraints, and then, okay, let's just add more shifts, right? So there's kind of, ad hoc reactions. And then, of course, you're making more cycles, so there's more space needed, more FTEs, more QC, more documentation, right? That all drives up the cost. And so the choices that we have choices, okay? And the choices that we make can either leave us at the effect of, right? Which is at the effect of circumstances, or we can take control and we have some say in the matter. So we can design processes that actually overcome the odds or the problems that we are facing, and therefore result in smarter processing and better solutions. So some of our conventional columns, while they serve us on a small scale, we'll say are fine, but really contribute to low flow rates, long cycle times. As you scale up, you have pressure issues and large space requirements. Okay? People encounter this, but they don't talk about it. They take it as a matter of fact. The fact is that you can develop processes and do processes in the lab on little height trap columns that are 3.5 centimeter bed heights. They work fine, right? But somehow there's a need to go to large bed heights when you go to conventional columns and scale up into manufacturing. And the choices of diameters and volumes don't necessarily match, right? Because they come in discrete sizes. And then conventional chromatography systems have kind of been the same. A few bells and whistles added to it. Maybe they can have little networking capability, etc. But the sizes are the same, the capabilities are the same. So that leaves you with having to do more of the same to solve newer problems that exist. And what about buffer management? Well, you can have large stainless steel tanks, so you can have storage totes, lots of plastic totes and flow kit connections, right? So it just compounds the problem. And so if you look at using these, what we call for Gen Zero antiquated technologies, what results is large footprint, large CapEx, complex workflow, more FTEs, higher labor costs. and of course, more consumables and slower process times, right? And all that, when you distill it down, maybe it's 8 to 10 batches that you can run in a year, and your cost, even if the titers are high, because people think that, hey, if we can get high cell densities, then we got our cost licked. Well, not so. You still have over 500 grams a liter, according to the model we worked with. Okay, and we'll see how that can be driven down to as much as a fifth, down to as a fifth of that. So, what are the 7 high impact choices that we can make? Well, believe it or not, the choice of column that you use is fundamental. It's a low-hanging fruit. It's very simple, and it can make a difference, significant difference in speeding up the process, reducing the footprint, and dealing with operational issues that result in process problems, right? The choice of the chromatography system. We'll get into some of this. Single-use systems. People are using single-use systems because, hey, now we can use multi-products in the same facility and amortize our facility costs over several different products. Good idea, right? And we don't have to do cleaning validation. We don't have carryover issues from one product or one batch to the other. Yeah. Now, if your titers were to go up and the scales were to increase, what would happen then? Okay, then I think people run into issues. Well, what if you had systems where you could have multiple size flow paths in the same system, right? And then what happens with the buffers that I use? What if you had inline buffer dilution capability? Okay. And then choice of making buffers. Well, the standard choice of making buffers is mixing tanks, right? Some people who are more evolved are starting to look at inline buffer blending. And what is that? That is taking your individual buffer stock concentrates. So a concentrate of an acid component of the buffer and a base component of the buffer. And so just basic chemicals, right? 1 molar monobasic phosphate or dibasic phosphate or acetic acid, sodium acetate, like that, along with 3 molar salt and Wi-Fi. Now, if you blend these together in the right proportion, you can get the buffer that you want. Okay, so there are these inline stock buffer blending systems that are available from some vendors, and people are now starting to use that and say, okay, now I don't have to have such big tanks. I can have smaller tanks, and I can make these buffers, but these buffers still have to go into totes, and then or either the buffers have to be made and pumped to some purification suite, or they have to be filled in totes and moved around. And then choice of batch or continuous. I think this is very fundamental. The FDA has given us guidance since 2013 to move towards continuous processing. Of course, there's a movement to bring more PAT, process analytical technologies onto the shop floor. And I think if as we'll see some of the data, that if you move from batch to continuous, there's significant savings in process times and footprint and reduction in cost. And then how about if you're running continuously, which some people are starting to do, they run again into other bottlenecks. So you solve one problem and you create another bottleneck, which is now you have to supply buffers continuously. And how do you do that? So, is there some potential of integrating that function into the continuous process? And then if you're running continuously, you've got multiple unit operations. How do you start to communicate with each of the other process steps and handhold? Are you going to have intermediate tanks and surge tanks in between? How are you going to take samples? What are you going to do about that? So I think those are all the right questions to be asking. So you get to the answer by asking the right questions. And those are the right questions that we are raising here. And we provide you with some guidelines or some of our thoughts on how those can be handled, okay? And what we are doing to handle some of those. So starting with the column, many of you may not be familiar with the technology of radial flow chromatography. So these are columns in which flow doesn't take place along the axis of the column, which means that you typically have long bed depths and very short areas, and therefore you can get you can get higher back pressures as you scale up. Even though you keep the bed height constant, you can't always keep it constant because it's not a one-to-one correspondence in terms of the increase in diameter to the volume that you want. But even having 20, 25, or 30 centimeter bed heights is not necessary. You can go down to 10 centimeter bed heights, but then the diameters become bigger, right? So that's a problem. So here, you have a column in which the flow is applied to the entire outside periphery of the wall. And instead of having frits on the top and the bottom, you have two concentric cylindrical frits, right? One on the outside, one on the inside, and the resin is held between these two frits. So when the flow comes in, it's applied to the entire outside periphery of the column and then travels horizontally in through the resin bed, through the inner frit, and out the bottom. So you have a very large area over which you apply your flow. And so for the same linear velocities, you can have totally a much higher volumetric flow rate. And now the bed distance is the annular distance between the outside and the inside, which you can keep very small, so your back pressures are very low. And you scale up simply by increasing the height of the column, right? The physical height of the column, so, or the length of the column, meaning you go up. And this has been, today, these have been proven across all the major plasma fractionation industry, and for making antibodies, for making various recombinant proteins in some of the largest scales in the biotech industry. They also use in the dairy industry, where we are processing over 1 million liters of milk a day through four, just in this case, 4 350-liter columns running 1 million liters of milk a day. So very high throughputs, doing ion exchange, right? Traditional SQ ion exchange, no big deal. And no back pressure issues, nothing. And these things have been running for years now. So even the large, and in this particular picture here, for instance, you see plasma fractionation plant with 4 350-liter columns, radial flow columns. These things can be automatically packed and with packing skids where you can write a protocol and they're packed, and the HETP asymmetry, the bed integrity is established and determined, and it's either pass or fail, and you can move the columns in line, right? And there's some data here from a company that we worked with that's well known called Genentech, and they show that with conventional columns, the process was taking 8 hours, and the same process with radial flow column was done in 2 hours. And as far as the product impurity profile is concerned, it was identical, right? So 4x improvement in throughput, very low hanging fruit, and all you're doing is transitioning from one column type, one hardware change to another. It's like using a better pump or using a better tank, right? You're not changing the CMC, you're not changing the chemistry, you're not changing the resin. So it's the same ion exchange, affinity resin, hydrophobic interaction resin that you can use, right? So again, what does that afford you? Well, it's not a big validation. 3 batches, show consistency, and you're done. Of course, you have to consult your CMC in the transition. We can advise you on some of that, right? Because we've done that across the board. We are doing a post-approval change on a 2.5-ton insulin plant right now, one of the largest in Asia, where they're moving to radial flow columns, and that gives that plant a 30% added capacity because that step was a bottleneck in the process, right? So you can achieve dramatic results depending on your process and opportunities. Sometimes it's an idea to just incorporate this right in the beginning so you don't, so the idea is to incorporate these kinds of tools right in the beginning so you don't face the problems once the process is tossed over the wall and goes into manufacturing, where it's later going to be scaled up and become somebody else's headache. So with that, I'll share with you a little video. I don't know if the sound is working or not. Yeah, Reinhard, you have a question? Yeah, so you simply increase the size, the height of the column, and then of course you get higher areas which are in proportion to the volume of the column, so you can go at higher flow rates. Right? I won't have you watch the entire video, but this is just to make a point. If you guys are interested, you can certainly go on YouTube or on our website and see these videos, right? So I'm going to proceed now. The choice number 2, right? So the first choice was consider radial flow if you're doing ion exchange, affinity, or hydrophobic interaction, because it's going to give you about 4 times the throughput of conventional columns with the same conditions, right? And even if you're concerned about, we could give you various strategies. For instance, you can do wash steps, solution steps, regen steps very fast, and do the load steps slowly, etc. Right? So there are lots of strategies around it. We work with all kinds of resins. The second thing is the choice of the scale, the chromatography system. We have systems that go from the bench scale all the way to large scale, and lots of unique opportunities there to make your process more robust, right? And also reduce the cost, reduce the footprint. So the size of these systems are very small, so you can get a high capacity in the same space because you're very conscious of that. And you have a very high turn down ratio of the pumps. And what does that mean, right? If your turn down ratio of your pump is 10-1, that means that you can only go, let's say, between 100 ml per minute and a liter a minute, or 10 liters a minute and 100 liters a minute. So if you're going much smaller on the lower end, then especially if you're doing gradients, right? Which may, if you're running at 20 liters a minute and you're doing a gradient, which is a 10-1, requires a 10-1, then one of the pumps has to go down to 200 mils a minute. There's no way that that's going to happen, right? With conventional technology. Now, with our systems, with 100-1 turn down ratios, you can actually do that. Is you can go down to much, much smaller flow rates and get higher degrees of accuracy. So the performance is there for both for smaller columns, as well as in the same machine, you can scale up to larger columns. Right? You don't have to ditch your machine and say, oh, well, you know what? Now our titer has gone up twofold, threefold. we need this bigger machine, right? Or we have to do some work on the pilot plant, and so we have to have a separate machine for that. You have wider range, and that gives you more options and in the same footprint, right? Lots more other advantages, but I'm just pointing out a few nuggets for you to take home with you today. Certainly engage with us if you're interested. Then what about buffer making? Well, most buffers are just made in tanks, right? You have tanks where you fill in Wi-Fi and throw in your powder, stir it, make your 1X buffers. Well, we now have capabilities, not just us, but across the industry from various vendors where you can, in the chromatography systems, you can do inline buffer dilution, right? So in our machines, that's available as well, where instead of having a 1,000-liter tank where you make 1X buffer, you could have a 100-liter tank, or you can make 10X buffers. You have to look at solubility, and things like that. But conceptually, you could make 10X buffers. And then the system itself would have the ability to marry in or blend in Wi-Fi, right, which is water for injection in line with that concentrate, and deliver the 1X buffer to the column at point of use, right? So strategy people are starting to use, good strategy. And then, of course, one step further than that is stock buffer blending. And what is that? That's what I just talked about a little earlier, where you actually have the chemical constituents in liquid form. So you may have acetic acid, 1 molar, or you may have 1 molar acetic acid, or you may have 1 molar sodium acetate. So, then 3 molar salt and Wi-Fi. And these can be blended together to get the buffer at the right concentration and the right salinity and the right pH. And there are systems available to do that. So we'll come back to that in just a minute. Choice number 3 is single-use machines, right? So there are single-use chromatography systems from the bench to the large scale that can be used. Now, if these machines have the ability to do this inline dilution, then you can simply take smaller totes instead of, 100-liter or 500-liter big tote. You could just have a bag, right, with 10-liter bag or 50-liter bag and connect it to the single-use chromatography system and then do the inline dilution. And in some of these machines, we can give a threshold, we can give a target conductivity, and give a threshold value. So we ensure it's not just some mechanical dilution and hope that everything works. No, we can ensure that the buffer meets that target concentration or a band around it, plus minus, whatever it is, 2 millisiemens, so that the buffer is actually delivered to the column within that specific parameter, right? And also in these machines, you can change out flow paths with different diameters. So in the same machine, you can scale up as much as a factor of 10, right? So you, for instance, have a system that does 0 to 300 mils a minute. And the same machine, you can put a bigger tubing flow path, a bed scale system, which can go to 3 liters a minute. You got 10 times the flow rate, 10 times bigger column that you can use. All the scale-up work, the development, as well as the pilot and the scale-up work can be done essentially for folks in gene therapy in one machine. So that's the kind of thing that we are talking about that saves on FTEs and saves on CapEx and gives you that added flexibility to be able to reduce costs, right? Time to market issues as well, because if you're waiting for equipment delivery to scale up your process, that's time lost, whereas you can do it in the same machine, you can move along much faster. And I'm not gonna, there is a video, but just in the interest of time, I'm not gonna play that. Again, if you guys are interested, go on our website, go online, and you can learn more about these kinds of systems. And we have a lot of such systems, right? From the bench scale, as small as 0 to 300 mils a minute, all the way to several thousand liters per hour, right? So different scales, the ability to do dilution, gradients, change different flow paths, etc. The flow paths come sterile. I'm not going to go into details, but just want you to be aware so you can make those kinds of choices. And here's the plant that we were talking about. We're doing 2.5 tons of insulin. This one, they decided to use radial flow columns and skids that had this inline buffer capability so they could reduce their tank volumes from 6,000 liters for making buffers to, or storing buffers to 2,000 liters. That's huge. And then they were able to get 30% more capacity in their plant, right? So just imagine, same number of people, same plant. If you can reduce the cycle time of the batch, right, the batch time, that gives that plant that much more added capacity, and that's that much more, that's that much more revenue output from that plant or reduction in cost per goods from that plant, right? Because you're making more, but with the same fixed assets that are there in the plant. Now, we talked about buffer making. Well, this is the traditional, we are calling this Gen 0 and Gen 1 and Gen 2. And let me explain this in pictorial form. So Gen 0, which is what most people have today, are a bunch of tanks on the left, on the top, where you make buffers, right? So you have different tanks, so you're making different buffers. Then you have these large totes, storage tanks, in which you're storing the buffer. And from there, after the buffer is made, you put them in these small mobile totes and move them to the purification suite, right? Where you have a chromatography system and you have a column. So these things have to be moved. These buffers have to be made. They have to be stored. And, our storage conditions are going to vary. The conductivities, pH, all going to be the same. With this time frame, with all these number of people and labeling and documentation that's needed, right? Just think about it. So the Gen 1 solution to this issue has been, this is from some other suppliers, where you have a machine, which some people call a buffer blending system or inline conditioning, where you can take these concentrates, right? You can take these concentrates of these buffers, individual components. So not concentrates of the buffer, not like a 10x concentrate of an equilibration buffer, but the individual components, the acid, the base concentrates of the acid and base components of the buffer, concentrated salt, Wi-Fi, like that, run it through this machine, and then punch in, whether you want a 0.05 molar phosphate with, say, 0.4 molar salt, right? And then it'll make that particular solution. Then you put it in these totes, and then you take them to the chromatography system and run it on the column. So certainly simplifies the workflow over Gen 0, right? There's capital expenditure in machines like this, but then you reduce all this capital expenditure around the tank and around the storage and all that. So this portion of the solution remains the same, but you combine these two into this machine, right? Now, Gen 2, which is what we are talking about, which is one of our elements of smart processing. We call it the QuantiCept downstream processing solution, is simply this. that you have these components of the buffer, the concentrated components of the buffer, and you have a chromatography system which has the ability to do the inline buffer blending from stock concentrates connected to Wi-Fi and transparent to you in the background. An operator from an HMI just calls up the equilibration buffer, gives the pH and conductivity, the duration of the run, etc., the volume, and runs it through the column, right? So now you've done away with all the tanks. You've done away with even the Gen 1 solution, which is this blending system and the chrome system here and these intermediate totes. And all you do is make the buffer, do the chromatography in one single solution, in one single machine, run it right through the column. See what I'm talking about in terms of process simplification. What does that do to your FTEs? Number of people required, right? You don't have people handling the tanks. You don't have people doing this stuff here with buffers. You don't have people running chromatography columns. The same chromatography operator can do everything, right? Reduces the cost, reduces the complexity, reduces the FTE, reduces the space. And here's some data. Some of this came about because of the work that we did with suppliers of COVID-19 vaccine, where they had to scale up to several 100 million doses a month, right? They didn't have the facility. They didn't have the space to make buffers. In a matter of 3 months, they needed to be producing large amounts. Here's a system that we made for a company that was running at 7,500 liters per hour. And we enable them to do chromatography with inline buffer blending, right? It was a complex buffer. It's not just a standard, acetate, phosphate buffer, but they had other additives, which I can't go into, added to it. So we had more pumps, and the pumps are being ratioed. But it was very simple. Nobody had to write down ratios of pump flow rates or anything like that. The operator simply wrote down what the molarities of the incoming solutions were that they were attaching and what the desired molarity and the pH and conductivity of each of the constituent solution buffers were. Whether it was a wash buffer they were making from the same components, or an elution buffer, or equilibration buffer, regen buffer, like that, right? So here you can see that we had 200 millisiemens salt, and we are making an equilibration buffer that's 7 millisiemens, right? So from a stock concentrate, and then the pH is 7.2, right? So in this case, of course, the wash buffer also had a pH of 7.2. It could be different. And then of course, the conductivity went up to 24 millisiemens, right? So, and these are in seconds, by the way. So in a space of 45 seconds, you reach equilibrium, right? And that's just the time it takes to get the new buffer, pumped through the whole piping and out to the column. And we establish a band in which that buffer is supposed to work. I mean, you're going to accept it. Until that point, the buffer exits the column to waste. And when it's within that range, it goes into the column, right? So very simple, but very impact making. And again, we have a video about that, just kind of keeping pace with, just in the interest of time, I'm not going to play this video. So I'm going to move on to the next topic. I want to be cognizant of your time. And so the next choice, so that's one choice, right? The choice to integrate buffer making capability into the chromatography skid itself. So you eliminate the whole business of buffer making, right? Imagine what would that do for your operation. And we are happy to talk to you and engage with you and point out what that impact could make in your particular scenario. The next high impactful choice that one can make is going from batch to continuous production. Now, some processes we understand are small volume, doesn't need to go to batch production like antibodies. But there's enough data out there that shows, see, on the right-hand side, enough models have been done, enough work has been done, that you can see the size of the plant for batch versus continuous production is considerably smaller. Amount of liquids that I use, amount of totes that I use, etc., shrink considerably, right? I won't read all these points to you, but the point of it is that, and of course, once the process is running continuously, you don't have a lot of transfer steps, hold steps. So number of FTEs goes down considerably, right? Number of disposable totes goes down, search tanks goes down, and the daily buffer consumption also goes down because you're making cycles, fast cycles, footprints goes down, amount of QC goes down, right? Like that. And this is, again, these are not our data. So I want to credit whoever has done this work, some very smart people, and showing that there could be as much as a 5x reduction in CapEx as you even moving from fed batch to continuous, right? From batch to fed batch, there's a reduction in cost and footprint. Again, from fed batch to continuous, further reduction. And you can be smarter in your processes by staggering things. You don't have to finish the whole process, right? You can get it started. You can get one batch started, and you stagger it as it's gone through one goalpost or one unit operations. You could start the other one. So you can do those kinds of things to increase productivity. But the ultimate, of course, is continuous purification, continuous production. And whether it's continuous upstream perfusion, in the case of mammalian cells, cell culture tied to, some form of cell removal, whether it's continuous centrifugation or an ATF type device going into continuous capture chromatography, or, and then flow through other steps beyond that, or you could have just large batches from fermentation and still have continuous downstream processing. And so in the area of continuous processing, Sepragen has systems going from the bench scale all the way to large scale, right? We have these in multi-use form. We also have them in single-use disposable flow paths, right? And so, and then we also, if you're going to do continuous processing, it doesn't make sense to make buffers in batches, right? It's obvious because you're just trading one bottleneck for another. And you just have a bucket brigade, right? Of buffer totes lined up, which you're changing out constantly as you're running out of buffer in a continuous plant. So then we have these bench scale systems. These can go, 250 liter perfusion, all the way to 2,000 liter perfusion, right? And we could do larger. So, and then we've also had these where we've integrated 2 or 3 column continuous operation where one column is loading, let's say the other is eluting, or you have 3 column where one is loading, the overflow goes to the second column, so you maximize the capacity of the first column, while the third column, which is saturated, then goes through the region wash and elution, like that, right? So, but what we've done is also integrated the buffer blending part into these systems. Very user-friendly, simple software, 21 CFR Part 11 compliant software to run these machines. The software is not islands, right? So they're not like individual islands. We can connect to these systems can be connected to your network. And so login is through the network. We can talk to your network through OPC UA, talk to connected processes upstream and downstream through Profinet. So we have a whole software team that supports that. So anyways, this just kind of gives you an idea about this. Here's a picture of that, of the operating screen, right? So here the orange is the load. You can see it's going on to the first column and then goes to the second column, whereas the third column goes through the non-load steps of washing, elution, regen, and equilibration. And here's what we were talking about with the buffer blending, right? So you have the acid component, base component, can have multiple buffers, right? So you have 3 acid components, 3 base components, 3 different salts, additives, Wi-Fi, like that. And going through, we've got flow meters on each of these. We can write recipes. The recipe editor is very simple, small here. We won't go into it in detail. There are videos online. You can certainly look at that, which explains how this is all done. And we can do a one-on-one in more detail and do a deeper dive, show you guys how simple it is to program these machines. But these can be available in single use flow paths like you see on the skid on the right. Right? So this is the buffer maker where different buffers are being made in line on demand, right? An equilibration buffer, the wash, solution, regen from the same components. These are not individually different pre-made buffers, but just the chemical constituents, concentrates, right? And you can run continuous. So what does this do? It reduces space, definitely, right? It reduces CapEx, it reduces the number of people, and it reduces cycle time. And in this particular case, really, when you look at that, it's the function of this machine then is a function of another vendor's continuous system and their buffer making system, right? In another case, you would need that continuous system, very complex piece of machinery, but you can run continuously, but then you'd have to make buffers, put them in totes and move them around. In our particular case, right, which is the Gen 2 solution that I've been talking about, all of that is integrated into one machine with one HMI. So an operator just makes the run, programs it, and you're ready to go. And this is just more data showing that, yeah, you can do continuous processing, you can do continuous buffer making. They're within the specs, very quick, elegant processes. And what does it do? Well, ultimately, if you look at it here, this is a Gen 1 facility, continuous processing facility, right? So this is, if you use a stock buffer making machines, you could have this concentrated buffer components, you have these storage totes, and then you can bring them onto a continuous line, okay? Now, with the solution that we are proposing, get rid of all this, right? Because the buffers are made here, you just have these tanks move out here and have the stock concentrates feeding these skids that are doing continuous processing and making the buffers. So so this is going from a Gen 1, they would call a Gen 1 solution, not Gen 0, where you have all the tanks and all that, but from Gen 1 to Gen 2, and simplifying the footprint, the process even further. And it's not just simplification for the sake of simplification. It has a significant impact on CapEx. So traditional design, which is a Gen 1 design that we had shown you on the left, for instance, might be for the process steps, right? Just the capital equipment would be 13.6 million. Whereas in this particular case with the Gen 2, it's 7.5, right? So you're not talking about facility costs and all that, just the FTEs and all that. You're just talking about the actual facility, downstream facility. And then the last one I'll say is your choice of concatenated processes, right? Which is that you can you have the capture system. This is a typical antibody. This, and then you can have these unit operations like straight through process, anion exchange, hydrophobic interaction, and UFDF, which are all integrated on one skid. So we can support you with that. It all results in a cost of goods of $130 a gram. So in our model, comes down by a factor of almost 5. Of course, the process is much more simplified, and we've done many systems like this, and we are happy to help you with these and other ideas to reduce your cycle time, footprint, FTEs, and cost of goods. So we have a full sort of customized engineering department with process engineers, with mechanical, electrical, software folks, and we can work with you on your specific design. You might think your processes are unique, but we've come across a lot of them and therefore have lots of experience. We've worked with A&E folks, A&E companies, and end users directly. Reach out to us. We have manufacturing capabilities, 3D printing, clean room assembly, all in-house, and we can support some of the largest projects in the industry fully with FAT, SATs, IQ, OQs, all the documentations and turnover packages. And I've worked with a large number of companies across the globe. So that's us, and I will conclude now. It gives you a little bit of an idea of what it is that we wanted to share with you, and I wanted to thank you very much for taking your time to join us this morning. I know that wasn't necessarily easy for people, but really appreciate your joining us and hope you learned something of value today. Reach out to us if you have any questions. And with that, I'll conclude. Thank you very much.